Namaste all of you. My name is Dr. Ritu Singh Rajput, Faculty of Agriculture and Veterinary Science, Department of Food and Biotechnology, JAYOTI VIDYAPEETH Women's University, Jaipur. Today in our session, we have discussed about the RDT. That means recombinant DNA technology. What is the recombinant DNA technology? We will discuss in this lecture. Recombinant DNA technology refers to the joining together of the DNA molecules from the two different species that are inserted into the host organism to produce new genetic combinations that are value to the science, medicine, agriculture, and the industries. Recombination DNA technology, on the other hand, it is the general name of the piece and for the DNA that has been created by the combination of at least two strands. They are DNA molecules found by the laboratory methods of the genetic recombination, such as a molecule cloning to the bring together genetic material from the multiple sources, creating sequence that would be otherwise found in the genome. Recombinant DNA in the living organism was first achieved in 1973 by the Herbert Boyer of the University of California at the San Francisco, at Stanley Cohen at the Stanford University, who used E. coli restriction enzyme for the insert foreign DNA into the plasmids. And how to it will be work? First, you have to take F plasmid ligate with the itself, then gene of the inserted ligate itself. Then in F plasmid is the electrocorporated with the cells and electrocorporated into the cells. Then after that the colonist turns blue, Z genes unbroken, and the cells die and no antibiotic residence. It will be available. General steps of gene recombination, which is the first of all we have to do isolation of genetic material. Second is the restriction enzyme digestion, then amplification of using of the PCR, and then ligation of the DNA molecules, then insertion of the recombinant DNA into the host. After that, we have to do isolation of the recombinant cells. First of all, we have discussed about the isolation of the genetic material. How do we isolate the genetic material? The first steps is the rDNA technology is that is called recombinant DNA technology is the isolated the desired DNA in its pure form, free from the other molecules. Since DNA exists within the cell membrane along with other macromolecules such as RNA, polysaccharides, protein, lipids. It must be separated and purified, which involve enzymes such as isoenzyme, cellulase, chitinase, ribonuclease, protease. Other macromolecules are removable with other enzymes or treatment. Ultimately, the addition of ethanol causes the DNA to the precipitate out as a fine threads, and it is then spooled out the give purified DNA. Second step is our restriction enzyme digestion. Restriction enzyme digestion act as a molecular scissor, and it can cut DNA and specific location. The reaction are called restriction enzyme digestion. They involve the incubation and purified DNA with the selected restriction enzyme, and condition are optimal for the specific enzyme. The techniques which will be used in agarose gel techniques, that is reveal the process of the restriction enzyme digestion, and the techniques involve running out the DNA and on the agarose gel or the application of the current. The negatively charged DNA travels to the positive electrode and it separates out based on the size. This allows separating and cutting out the digested DNA fragments. and the vector DNA is also processed using the same procedure. And the third step is the amplification using PCR, which is PCR is the general method for making multiple copies of the DNA using the enzyme, which is DNA polymerase in vitro. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. PCR reaction run into the thermal cycles in following. First of all, we have to make template DNA to be amplified in primers, sample chemically synthesized oligonucleotides that are complementary to the region of the DNA, and the enzymes which is DNA polymerase using the PCR, and the nucleotides needed to be extend the primer by the enzyme. After that, the cut fragment of the DNA can be amplified using PCR, then ligated with the cut vectors. Our fourth step is the ligation of the DNA molecules, and the purified DNA and the vector of the interest are cut with the same restriction enzyme, and it gives the cut fragment of the DNA and cut vector, and now it is open. The process of joining these pieces together using by the DNA ligase, and the resulting of DNA molecule is hybrid of the two DNA molecules, the interest molecule and the vector. It is a terminology of the genetics, that is intermixing of the different DNA strand is called recombination. Hence, it is the new hybrid molecule is called as a recombinant DNA molecules, and the technology is referred to the recombinant DNA technology. Our fifth step is the insertion of the recombinant DNA into the host. In this step, the recombinant DNA is introduced into the recipient host cell, mostly a bacterial cell. The process is transformation, and the bacterial cells do not accept foreign DNA easily, and therefore they are treated to make them component to the accept new DNA. This process used may be thermal shock by the calcium ions treatment or the electroporation. Our sixth step is the isolation of recombinant cells. The transformation process generates a mixed population of the transformed and non-transformed host cells. The selection process of the involved filter the transformed host cells only for the isolation of the recombinant cell or the non-recombinant cells. Marker gene of the plasmid vector is generally employed in isolation of recombinant cells. For the example, PBR322 plasmid vector contains different types of marker gene, which is ampicillin resistance genes and tetracycline resistance genes. When PST first and PST first RE is used in a knockout ampicillin resistance genes from the plasmid, so that the recombinant cells become sensitivity to the ampicillins. Now we have talked about the application of the recombinant DNA technology. Recombinant DNA technology widely used in the biotechnology, medicine, and the research. The common application of the recombinant DNA technology is the basic research, in which technology is the important to most current work in biological and the biomedical sciences. RDT is the use to identify, map, sequence genes and determine their functions. Recombinant proteins are widely used in a regions in a laboratory experiment and generate antibody probes for the examining protein synthesis within cells and the organism. Many additional practicals application of the recombinant DNA are found in the industry, food production, human veterinary medicine, agriculture, and the bioengineering. DNA technology is also used to detect the presence of HIV in a person. An application of recombinant DNA technology in agriculture, for example, manufacture of the BT cotton to the protect the plant against bollworm. And we have talked about the application of the medicine, insulin, and production by the DNA recombinant technology is the classic example. In gene therapy, it is used as an attempt to correct the gene defects which give the rise to hereditary disease. And we have talked about the clinical diagnosis. ELISA is an example where the applications of the recombinant DNA is possible. Now we have to talk about the limitation of recombinant DNA technology. Destruction of the native species in the environment, the genetically modified species are introduced, and the resilient plants can be theoretically given rise to the resilient weeds, which can be difficult to control, cross combination, contamination, and the migration of the proprietary DNA and between organism, recombinant organism contaminating the natural environment, and the recombinant organism are population of the clones variable in exact same ways, and the single disease or the pest can be wiped out from the environment population and so fastly, and creation of superbug is hypothesized, and ethical concern about the human trying to the play God and mess with the nature's way of the selection, and exaggerated by the fear unknown of the what we have can be created using the technology, and how is it the going the impact of the civilization. Such as a system uh, might lead to people having their genetic information stolen and used it without permission, and many people worry about the safety of modifying food medicines using recombinant DNA technology. So these are the some kinds of limitation which is in occurred in recombinant DNA technology. And in a recombinant DNA technology, cloning is the most important part of this. And what is in our previous lecture we have discussed the what is the cloning, and vector is also important part of this. Just because if we have not particular vector, we can synthesize our particular DNA or particular samples. So if we have doing a vector is important, their size should be small enough and separated from the chromosomal, and it should be proper plasmid, and it should be had some site of the ori which is responsible for the DNA replication, and it has some multiple cloning sites which is called as the MCS, and it is also have a selectable marker genes and allowed to the select for the transformed colonies, and it has also some RNA polymerase promoter sequence, and it has used for the transcription in vitro and in vivo. It has some DNA sequence primer. So vector is also important for this. And there are some kinds of vector. There are so many types of vector, which is the bacterial plasmid vector. It can be clone inserted that the smaller than the 7 kb. Some express in eukaryotic proteins from the gene poorly, and some are bacteriophage vectors, and some are cosmid vectors, some are the expression vectors, some are the bacterial artificial chromosome which is called BAC, some are yeast artificial chromosome which is called YAC, and some are TI vectors. So these are the good vectors which is type of required in cloning in recombinant DNA technology. So, and vector is also important role. They should be provide some cosmid vectors. It has contained cos and lambda DNA. They should be have best expression for the bacterial expression vectors, and they should be assumed for the human insulin using the bacterial expression vectors, and some now I will tell you some disadvantage, which is the for the how do we identify the clone of the gene interest, which is the if we have introns and are cloned in the addition to the exons, some majority of the genetic DNA is introns in eukaryotes. So majority of the library will contain non-coding pieces of the DNA, and many organisms have very large genomes, so searching of the gene of the interest is difficult and it is time consuming. From this RDT, we have to make easily cDNA libraries, and it is also important just because we have to make a small mRNA from the tissue interest is also isolated from the cDNA libraries, and there's so advantage of the cDNA libraries. And colony is also hybridized in through, and PCR also play important role in this. And some techniques are also using in RDT, which is the PCR, restriction site, restriction mapping, and southern blotting, northern blotting, and electrophoresis, and for the gene expression, bioinformatics also useful part of this. So bioinformatics also is important part of this by the Human Epigenome Project, International Harpman Project, Encyclopedia of DNA Elements Project, Personalized Gene Projects, Cancer Genome Projects. So these are some genomics which are including in helpful in the discipline of biotechnology. And this is all about the RDT, and I have give you the proper scenario and proper introduction of the RDT. And if we have talked about the exam point of view, generally they will ask what is the RDT, and just tell about the some techniques which will be using in the RDT, and what is the advantage of the RDT, and what is the some kind, explain the disadvantage of the RDT, and some application, explain the some application of RDT, and what are the limitation of the RDT. So these are question arrived in exam generally. Thank you so much.